So I'm Ron Board. I'm the tree nut breeder at the Center for Agroforestry. My goal here today is to give you a brief overview of the chestnut improvement activities going on at the center here. So some brief context, a cultivar collection was established in 1996 at the center with over 60 cultivars maintained today. The majority of these originated from growers and enthusiast importations and evaluations over the 20th century, including many from Greg, where OP seed was from grower selections was frequently circulated. In 1999, a subset of 12 promising cultivars was established in a replicated trial at MU, leading to the first recommendations for the Missouri River Hills, which is an ideal environment for growing chestnut. Because of delayed graft failure, incompatible interactions are seen to the right here. And interest in cultivation across a wide geographic range, growers outside of the Missouri River Hills opt to plant seedling orchards, which are often planted around a double density so that poor performing seedlings can be culled. To support the success of seedling orchards, UMCA has distributed OP seed from the cultivar collection to growers and nursery partners like Greg since 2008. Today, I want to overview activities 4 through 6 listed here to characterize on-farm selections, establish a new replicated selection trial at the University of Missouri, and organize for next generation on-farm breeding through the Chestnut Improvement Network. To highlight some breeding goals in a quick snapshot, there's a focus on stable production by environment, which includes but is not limited to late leafing and frost avoidance, as well as appropriate harvest and leaf sinensis times to avoid late fall cold damage. Of course, high kernel quality is important, like size, color, texture, and flavor, and a low rate of defective characters like split shells or off flavors. There's a common carrot-like flavor in some cultivars. So these are also important performance characters, a high crop load through increased burrs per shoot or male sterility, and then resistance to a variety of diseases and pests. And I know Greg could probably relate with the statement that could sometimes seem like a moving target. Okay, so on to characterizing on-farm selections. So over 1,000 growers have received UMCA repository seed. This map gives you some insight into the extent of seed distribution over time. The depicted farms, blue and red dots, receive seed either directly from the UMCA or a local nursery partner, but this map doesn't even include many of the growers that Greg has distributed seed to, for example. The seedling base represents an invaluable resource to select improved offspring in diverse environments. So we seek to build our improvement efforts from these materials. Participants in on-farm evaluations that we've conducted in my lab since 2020 are seen by the red dots. Evaluations have grown to over 30 growers and around 600 selections across 11 states and are ongoing. Evaluation goals include phenotyping on-farm selections for traits of interest, discerning on-farm selections ancestry and parentage using a DNA marker set developed by collaborators at Notre Dame, utilizing data from these first 2 objectives to construct core collections for conservation and breeding. And then lastly, capitalize on momentum from these evaluations to organize a participant network as a new breeding initiative to begin next-gen improvement. So a little bit on the genotyping, many cultivars of Castanea mollissima, the Chinese chestnut, or many cultivars are Castanea mollissima or Chinese chestnut complex hybrids with various other Castanea species. Interspecific ancestry is common due to the dynamic history of sharing OP selections over the 20th century. The Romero Severson lab used a training panel, which is the structure output you see here, to develop a set of 45 EST-SSR markers capable of discerning complex ancestry amongst Castanea hybrids. We don't have time to discuss the data here, but the UMCA repository has been genotyped to confirm or correct ancestry And data from on-farm selections, so the 600 selections, is now being analyzed to do the same, as well as confirm parentage. And we expect these data to help us draw insights on parental combining ability and possible beneficial interspecific ancestry. So on to phenotyping, we won't take a deep dive, but this slide gives you a good picture of the phenotyping workflow. There are 35 characters in total. 7 are qualitative agronomic characters that we take in the field, including a visual assessment of crop load, number of burrs per shoot, and canopy architecture. You could see male sterile and male fertile individual at the top of the slide. Nut and kernel quality includes 16 traits, some of which you see here, and these were typically taken on 2 replications of 15 nuts. Spherical nuts are preferred, and an important size threshold is the 11 to 12 gram range. Some markets prefer larger nuts. In tile F, you could see a partial pellisole detachment with a portion adhered to a kernel invagination, which would be a defect, although that's a pretty good pellisole removal. In tile G, you could see chestnut anthracnose incidents, formerly called blossom end rot. Greg could probably talk at greater depth on that. And so based on 2-year data, 40 preliminary selections have been made from the 600 selections under evaluation. So this is around 6%. These selections represent 10 sites. These sites are shown in red on the slide. 24 sites in green are still have selections under evaluation. Emphasizing nut size, crop load, and defect rate adequately narrowed the selection pool to a manageable preliminary set for propagation. But of course, pest incidents and other characters were considered too. These selections represent the beginnings of a breeder's choice set from this work, but require more evaluation, including replication with all selections in the same environment. So last fall, a completely randomized replicated trial was established at the Horticulture and Agroforestry Research Center. 5 grafted replications of the 40 on-farm selections and over 60 new entries from a collaborator, James Nave and Greg Miller, were also included in the trial. Commercial checks for long-term evaluation were included, which include the listed cultivars, but then many more. And the trial also serves to conserve the selections long-term, where eventual orchard thinning will not reduce the unique genotypes. So this will basically transition to a repository. Some of the selections will feed into the breeding pipeline as next generation parents, but in the interim, we've deployed hand-pollinated full siblings from our repository based on complementary hybridization, as well as select half-sibling families in balanced quantities so that we can have some assurance that we're systematically using our founder parentage before we move on to using some of these selections. So after the first year of progeny deployment, with the types of materials I just mentioned, we have 9 breeding members, which means the grower has planted 250 seedlings or more, typically of 5 families with at least 50 seedlings each. And then we have a few larger plantings in the northern Kentucky area, as you can see by the circle size, including a really exceptional planting of full siblings that represents a partial diallele design using around 20 founder parents from our repository that we plan to use for marker trait association work going forward. So I'll wrap it up with this slide. As we incorporate new on-farm selections and deploy new breeding generations across diverse environments, germplasm tracking and data collection will be powered with the management software eBrita, which is a Dutch software that Greg helped pick out. The software provides invaluable tracking from nursery to field establishment so that we can maintain seedling identity with QR codes associated with unique pedigree ID identifiers. So each seedling has its own pedigree identifier. This function is also applicable to in situ and ex situ conservation as well, I'll point out. And then when the trees come of age, so you can see how we're labeling seedlings in the nursery before deployment, but when the seeds come of age for phenotyping, we can refresh labels with more permanent labels, like you see on the cattle tag, to ease data collection and archiving across remote locations. So university researchers can create and upload data collection sheets within eBrita, like you see in the upper left-hand corner of the slide here, to make them available remotely or accessible anywhere using the web or web browser or field application. And then data can be directly entered either by a grower or remote field technicians into the field app, which automatically uploads or syncs the data to a cloud-based storage when returning to Wi-Fi. So I'll just say a quick thank you and note that we have a new website at the bottom of this slide.